All right, everybody. Welcome to the next podcast. This is podcast 3, and this is part of your ecology unit, which hopefully everyone finished over the summer. And what this deals with essentially is the lab concepts that are around the lab that we do as part of this unit. AP Bio Lab 12 is a lab that deals with a topic called productivity. It's called Lab 12 because it's the last lab in the AP Lab manual of the 12 required labs. Now, before we can really talk about what we're doing in Lab 12, we have to understand this basic term, and that is productivity. What do we mean in ecology when we are talking about productivity? But before we even address that, we have to address some other things. We need to know a couple other concepts before we know what productivity means. And the first one of those is photosynthesis, and the second of those is cellular respiration. And just an aside, do we know what this critter is right over here? That is a mitochondrion, and it is critical in this process right here of cellular respiration. And we will talk more about that as the year goes on. But for right now, you really should start associating this organelle, this cell part of eukaryotic cells, with cellular respiration. It is a mitochondrion. Okay. Now, let's talk in some detail about photosynthesis and what that is. I know everyone knows this. You've been taught this since you were very small people, but let's remind ourselves. Photosynthesis is the process of using light and carbon dioxide from the atmosphere and water to create food. Okay, and we are going to use the term food interchangeably with these other terms: organic compounds, sugar, we'll use that term a lot, glucose, which is the specific sugar that is created as part of the process of photosynthesis. All these terms are interchangeable with the term food. Okay, plants are creating sugar, glucose, organic compounds, whatever you want to call it. That is what they are making when they use light to do something a little bit fancy with carbon and water. Okay, they use it to build these organic molecules. Okay, now this is a really, really important thing for you to know, and we are going to hear this equation again and again and again all year long. You really need to know it. Now, this is showing you a nice balanced equation, and me not being a chemist, well, usually I end up leaving off the nice balancing numbers off and focusing on what you see here in the black, what is really just the compounds, qualitatively speaking, that are being used. Your CO2 is going into this equation. Plants are obtaining it from the air, also obtaining from their environment. Usually, if we're talking about land plants, from the soil. They are obtaining water, and they are using the energy of light to force these things to create that molecule, which is the glucose, C6H12O6, one molecule of that. And as a byproduct, some stuff is also given off besides the thing that the plant really wants, and that's the food. Oxygen gas is given off, and some more water is given off, not quite as much as was taken in. So, all of this over here is being used to make this one molecule of glucose, this biomass, this plant, its living body, its tissues. And we're also giving off some other things. And as animals on planet Earth, we are especially thankful that plants not only make food, but they also make that oxygen, without which we couldn't survive. Okay, so what is photosynthesis? What is cellular respiration then? Okay, cellular respiration is the fundamental flip-flop of that photosynthesis equation. Almost exactly. It is using oxygen as a tool to break down glucose, food, and as we break that glucose down, to obtain a usable form of energy. Okay? Now, this is the equation for that, and it should strike you pretty immediately that this looks really, really similar to photosynthesis, but it's just flip-flopped. Okay? In this case, the body organism is taking in a molecule of glucose. Okay? It is combining that with oxygen, and it is also using water. Okay, and we are getting the thing we want from that is to use this oxygen almost as a hammer, and we will talk more about this later, but to help us break down the glucose molecules, because it's got a lot of energy in it, because that's what we are hoping to get out in cell respiration is energy. We are trying to obtain usable energy. This molecule has lots of energy in it, but it is stored in the form of chemical energy, which is not terribly usable. We need to get that out. So we use the oxygen to help us break this down, and we get usable energy that we want. We also end up releasing into the atmosphere some CO2, and we also get some additional water. This is that photosynthetic equation flip-flopped. Okay? What that should imply is that these two processes are really dependent on each other, and we'll talk more about that in just a minute. But let's first talk about who does these processes. And again, I trust you know this. Who does photosynthesis? Well, of course, plants do. But so does algae, which is not a plant, not even in the same kingdom. Algae is a protist. Now, some people argue that some forms of algae should be shifted over to the plant kingdom, but they are still considered protist. And even some bacteria perform photosynthesis. A type called cyanobacteria. It used to be called blue-green algae because of the kind of color it would give off. But nonetheless, lots of things do photosynthesis on planet Earth. Who does cellular respiration? Well, even more things. Who does it? Plants. Wait a minute. I thought plants did photosynthesis. Well, of course, they do both. Because why? What are we doing in photosynthesis? In photosynthesis, we are making food. Okay? But in cellular respiration, we are breaking down food. Okay? So, essentially, in this step, the plants, the algae, the bacteria, whatever, make their own food. They make the sandwich. They go to the refrigerator within their own bodies. Their photosynthetic organelles make, except for in the bacteria who don't have organelles because they're bacteria, but their photosynthetic components make food. But those food molecules are of no use to them until they bust them up and get the energy out of them. So they use the sunlight to make the food and then break down the food to get the energy out of it. Seems kind of, a long way around to get to that energy, but that's what they have to do. Now, who else does cellular respiration? Well, of course, more other kinds of bacteria. These, interestingly enough, are strep bacteria that might give you a throat and infect your throat or whatever, but they use respiration. They're called aerobic because they're using oxygen. They are breathing, if you will, to get their energy, just like we might. I'm not coming up. All right. Animals, of course, do cell respiration. We're certainly used to that. We think of us doing eating food and breaking things down to get energy all the time. So that's nothing new for us. And Fungi do cellular respiration. We often think of them as plants, but they are not. They just act like plants. They sit with their little hyphae and whatnot embedded in the soil, but they are not doing photosynthesis. They are decomposers. They are breaking down the soil, the logs, and things like that, and extracting the molecules and then breaking down those molecules to get energy out of them. Of course, amoeba and other protists, other animal-like protists at any rate, not the algae, but these also, well, the algae use cellular respiration also, but amoeba cannot do photosynthesis. They just do the cellular respiration. All right, so essentially, only a few folks are able to do photosynthesis, okay, on planet Earth. It's these guys, these green guys, okay? Whereas almost everyone can do cellular respiration. Not everyone. There are some bacteria that cannot do it. They are anaerobic. They cannot do cell respiration. They have to do another type of chemical process to extract energy from food. But cell respiration is common. Plants have both photosynthetic organelles and mitochondria, so we need to keep that in mind. Okay, so fundamentally then, this relationship between cell respiration and photosynthesis is really, really fundamental and really important. The products of one process are the reactants in the other. The products of this process, okay, are used, well, byproducts at any rate. The CO2 is used here, the glucose and the oxygen are used back there, okay, as part of cell respiration. Now, this image again is just those two equations, and we are seeing hopefully the real true nature of this side essentially being this side of the equation, and this side over here being this side of the equation, essentially. Of course, what's different is the use of light in photosynthesis and the result of usable energy in cell respiration. Okay? Okay? So, what is productivity then? We know all about photosynthesis and cell respiration. How does productivity relate to that? Okay? Primary productivity is the better term, and when you hear primary in terms of ecology, the first thing that should come to your mind is producers. Primary producers is a phrase used very, very often. And primary productivity then is simply referring to the amount of actual light energy from the sun that actually manages to get converted to chemical energy, meaning food, organic compounds, plant tissue, whatever, into stuff by the autotrophs in an ecosystem over a period of time. Okay, so how much are the autotrophs able to actually convert from that sunlight into actual stuff, organic compounds, leaves and bark and the stuff of the plant? Okay. Now, notice that's not talking about really what the plant is using up itself just to do its life processes. What we want to talk about is what is actually physically apparent in the ecosystem. Okay? Now, by measuring productivity, scientists can do a lot of things. They can compare different ecosystems to each other, really wildly different ecosystems like this desert to this rainforest to this ocean, which has been done, and we can kind of tell what's productive and what's not as productive. But we can also compare the same types of ecosystems, but that may be different for other reasons, like, for example, a polluted pond or lake versus a clean pond or lake. Okay, and we could compare their productivities to see what we could learn. Or we can compare just one ecosystem, perhaps this particular pond, from one point in time on this day to another year down the road on a different day, and how productive is it being. So, how exactly do we measure productivity? Well, because there's kind of lots of stuff happening in this photosynthetic equation, we could have lots of ways we could measure productivity. We could measure how much carbon dioxide is being used up over time. We could measure how much glucose, how much plant material is being made over time, and we can also measure how much oxygen is being made over time in a given sample or ecosystem. We could measure water, but as you can see, water is appearing on both sides, and water is kind of a difficult thing to measure sometimes, surprisingly, not always, and really depending on the type of ecosystem you're looking at. But what we will do in this lab is we will be focusing on an aquatic ecosystem, which really does make water a very difficult thing to measure. Lab 12 does focus on aquatic ecosystems. So we need to use some kind of way to measure productivity that works well in water. And one of the easiest things we can measure is something called dissolved oxygen. They make little probes that you can see my students holding in their hands, and you can stick those probes in the water and measure how much oxygen is present very easily. So what is dissolved oxygen? Dissolved oxygen is oxygen gas, just like what you're breathing in this atmosphere, O2, but that has been dissolved in the water, is being carried in the water. Okay? Now, how does this oxygen in the air get dissolved in the water? Well, there's lots of ways that can happen, but generally speaking, photosynthesis puts oxygen gas in the water, and right here you see some eelgrass, very common in our Chesapeake Bay ecosystem. For example, that puts oxygen, as it does photosynthesis, it releases oxygen gas and puts it directly into the water. Another way to get dissolved oxygen in the water is by agitating the water. If you have a windy day and you're kicking up the waves, you can see these waves are turning up in the air, and they are catching water, and the bubbles of the water are being of oxygen are being forced into the water, and so that will increase the oxygen in the water as well. It's why we aerate aquariums. In my room, those pumps are causing the bubbles to go into the aquarium water so that the fish and crayfish and whatnot have plenty of air to breathe. Okay. Now, if we're putting oxygen in the water, it's a living ecosystem, and organisms need oxygen to live. It stands to reason that oxygen leaves water as well. What kinds of things remove dissolved oxygen from the water? Well, mainly critters, okay? Lots of critters. Fish that we think of. Of course, we already said the plants are using it too. Of course, they're putting it in. Bacteria that are not photosynthetic, decomposing bacteria, will take oxygen out of the water, and that's really critical. Now, do other factors as well, besides the living things, affect the dissolved oxygen concentrations in water? And certainly they do. For example, temperature can affect aquatic ecosystems. Warmer water cannot hold as much oxygen as cold water. If you think about it, the molecules in warm water, warm molecules are moving all the time rapidly, and so that tends to drive oxygen gas out of the water, and warm water tends to not hold as much, whereas cold water, the molecules are more still and are held more tightly or not driven off. Okay? Salinity can affect oxygen. As we already said, agitation. Depth, why might that be? Think about that. Okay, now, what we will do in Lab 12. We've got two parts to this lab. We got 12A, where we are going to compare warm versus cool water and just determine how much oxygen it can hold. That's really not productivity. That's just pure water, warming it, cooling it, seeing how much water oxygen is in the water. And we're also going to measure the effects of agitation on oxygen content of water. 12B is going to actually look at productivity, meaning we are going to have actual water from a pond that has hopefully living critters in it, and we are going to measure how much dissolved oxygen is in that water after we do a little something to the water, and that is going to be holding it in different amounts of light overnight. And so we will measure the dissolved oxygen that is produced in each water sample, and that will tell us how productive the water is based on how much light is there. Okay, so 12A, here's what we will be doing. You are going to be in the classroom. This is a quick and easy lab. You're going to get samples of cool water and warm water, okay, which I will have for you. You're going to record the temperature of each in your lab handout. You're going to determine the dissolved oxygen levels of each by sticking the probe in each one. Very simple. Record those dissolved oxygen amounts. Then you're going to get a sample of room temperature water that I will also have sitting, and you're going to determine how much dissolved oxygen is in it, and then you're going to pour this small sample into an empty milk jug, and you're going to shake it for several minutes. I can't remember how long you're asked to do it in the lab, but you will read that. You'll shake it for a certain amount of time, and then you will simply measure the dissolved oxygen again in that water sample to see if agitating it altered the dissolved oxygen. Okay, so that's just kind of getting you used to working with the equipment, measuring the different parameters, and thinking about the different things that can affect dissolved oxygen concentration besides excuse me, the things that are living in the water. Now, in 12B, we focus on productivity, and this is a little more complex. We have a few little equations that you've got to plug in. They're not difficult, but there's a little bit more going on. We're going to get samples of pond water, and we will measure how much dissolved oxygen is in our pond water at the beginning, and then we're going to expose each sample to a differing amount of light. So which variable is that? We've got a choice of dependent and independent. What is this? This is what we're altering at the beginning. So we're going to expose some to no light, some to lots of light, and some to a medium amount of light. And then we'll wait 24 hours, and then we will measure those same samples again and see how much dissolved oxygen we've had. What if we have more dissolved oxygen than what we started with? If we do, then we know that the producers are going crazy and they're producing lots of oxygen, more than any consumers in the water can use. And that's a pretty productive ecosystem. What if we have less dissolved oxygen than what we started with? Well, in that case, we know that our producers aren't able to crank out the oxygen, aren't able to crank out the glucose. They're not being very productive, and our consumers are outstripping their ability to produce oxygen. And what if they are exactly the same as what we started with? Well, either we know that the rates are essentially the same of production and consumption, or there's nothing in the sample to start with, and there's no living things in there. Okay, so that's what we'll be doing. We'll talk a little bit more about exactly how. I'll give you a little demo of how we're going to use the lab equipment, and that's it. So thanks for watching.